So, can you walk me and our listeners through this process? Where does it begin?
I like to think of methylation kind of like a clock. So at the 12 o'clock position, you've got methionine. So methionine is a protein that we typically get through our meats. And from methionine, it interacts with an enzyme in our body called S-adenosyl methionine synthase. Just a fancy word for an enzyme that creates this issue or creates a process in the body that breaks down methionine to where it can add a methyl group to SAMe, and then from SAMe it becomes the greatest methyl donor in our body. And SAMe then helps produce that DNA. It helps produce the RNA, the neurotransmitters, and everything else that goes on.
So, SAMe is kind of like a delivery truck handing out methyl groups.
That's right. So it is giving off these methyl groups, and then from those methyl groups, it turns into homocysteine. And from homocysteine, it either gets broken down into cysteine. So that SAMe, if you're thinking of the clock, you have methionine up at the top, you have SAMe at the 3 o'clock position, and then at the 6 o'clock position, you've got homocysteine that either gets flushed out of the body and turns into cysteine, or at the 9 o'clock position, homocysteine combines with folate and methylfolate and methylcobalamin in order to turn back into methionine and start that cycle all over again.